Okay, so for some of you just tuning in, one of the aspects that we had said, you did have a paper that was signed yesterday. That will not be due on Thursday. I'm just looking ahead that the numerical day of Tuesday is the 4th, I think. Okay, because up here it's hard to add 7 to 29 because I think it is the 4th is next Tuesday. Okay? Because yeah, see, that's just looking ahead. That's why it says that, so then I typically don't forget. So 7th, 6th. Oh, Tuesday will be the 5th, not the 4th. Okay. So let's perhaps shoot for maybe the 6th. Let me look to see once where we're at in our materials. That would be Wednesday. Okay. And we are halfway through. Let's see once how far we get. Then we'll have a little better clarification on where we will be proceeding next week. All right. But again, you still have your vocabulary evaluation on Thursday. I think it's just best to get it out of the way. Then you don't have to worry about it over your long weekend. Okay, what is everything that you can tell me about the phospholipid bilayer?
It's certainly something that exists.
Part of the cell, something to do.
Okay.
I will also add on to that.
I got none of that. Glycerol? It does. In other words, it keeps certain materials in and other materials out because there are different ways that materials get into your cells, and that's going to be the movements across the cell membrane, which we wrote them down. We just haven't discussed them yet. Can you name one of them?
Osmosis?
That's one. That's two. There's another diffusion. That's true.
Controlled diffusion?
Facilitated diffusion.
Like an something.
Well, it is, but those all fall under a category of what we call active transport because you're talking about the endo and exocytosis. That takes energy as well. That's how active transport is just a little different. For those of you who do move on to take anatomy and physiology, it means a little more when you start applying these very basic concepts to your nervous system. Because what happens is, I always got to think about this every time. It's called the sodium-potassium pump. You think after this many years that we remember that. What happens is potassium is allowed to diffuse, in other words, no energy, down its concentration gradient, but sodium is pumped uphill against its concentration gradient. And it's at an unequal rate, like 3 and 2. And when that happens, that's what allows that voltage difference for a nerve signal to transfer down along its, it's called an axon. Okay? There's a lot more that goes into it, but we're not going to go that deep until you take anatomy, which is a year away. Okay, so this is one of your basic cell structures. One of them is the nucleus, as you can see, and then these are all just different types of cell structures, okay? But as far as what all cells have in common, especially eukaryotic cells, how are those different than prokaryotic?
Or they're like simpler? Or is it the other way around?
Prokaryotic are much simpler because they lack something. Well, they're all cells. Yeah, prokaryotic organisms are unicellular. One's got a nucleus, one doesn't. But which one's which? That's correct. Okay? And oddly enough, it's just kind of something that really doesn't make any sense in our bodies. Red blood cells, they lack a nucleus as well. There's a reason for that. Okay? Because what do we know? What do you think the function of a red blood cell is? We only said it in class. We never wrote it down. It carries something throughout the body.
Protein?
No.
No.
With carbs?
No.
Energy.
I don't know.
Oxygen.
Hey, I actually got
Okay? It carries oxygen around. So what happens is the nucleus is just something that takes up extra space. So you might be asking, well, how does it actually replicate if it doesn't have a nucleus? Well, there's genetic material in there. Not a nucleus, but just has genetic material, if my interpretation is correct. So then, again, it allows you to carry more oxygen throughout your body. Okay, so a nucleus is one of the basic, always want to associate, there's three main structures that all cells have in common. It's not so much this because these will vary. They all will have a nucleus if it's an eukaryotic cell. Okay? Muscle cell, nerve cell, okay, skin cell, you could say liver cell, pancreas cell, all of them are going to have specific functions to do. And with that, it's going to have the directions encoded inside of it on what that cell's DNA is. Okay? Are we past this?
Yes, we're
Oh, that's why we're, okay. And then what this is, when we get to something called protein synthesis, okay, we talk about more of the DNA further in depth. Okay. So what's that plant process where they make food for themselves? Okay, so when you look at the basic term, photosynthesis, what do you think synthesis might mean? Exactly. So it's producing something by what when you say photosynthesis?
Absorbing light.
Okay, you're taking light and you're converting that into energy. That's where the synthesis comes from. So a plant will make energy for itself because of photosynthesis. The light has something to do with it, and you're making the energy. So what do you suppose this means? Just like photosynthesis, you're making something. So in this case, what are we making?
Proteins for the body.
Proteins. That's right. Now, this gets to be perhaps maybe a little more confusing when we look at this. It has a very similar function to what the endoplasmic reticulum is because they're both folded sacs, okay? But how I like to look at this and maybe explain it is think of a distribution center like Amazon or UPS or something to that effect. How is it that a computer knows to send a package down to Mesa, Arizona? We're talking about that earlier.
It's got like a code written onto it to tell it what it's
Yeah, it's got a 5-digit code. What is that? It's a zip code, okay? So if my interpretation of this is correct, a laser will read that and know to send that to Mesa. This one's going to San Francisco. This one's going to Tempe, Arizona. This one's going to Portland, Maine. So as it's going down the conveyor belt, the laser reads that and then collects that and modifies it. Okay, this is the plane for Portland, Maine. This is the plane for or the dump off for Mesa, Arizona. This one's for Tempe, Arizona. Okay? So what happens is your Golgi apparatus will collect these proteins and just modify them a little bit to send them to where it is they may need to be made inside the cell, because each individual cell is going to make a certain number of proteins for whatever it needs to do. Some of your very fast twitch muscles, those that are work really, really hard, Exactly, or they're found in your eyes. So you're going to have lots of ribosomes in there. Skin cells, probably not as much metabolic activities take place in your skin cells. So you think there's a lot of ribosomes there? No, there wouldn't be. So a liver cell is something that would have to work really, really hard. So it probably has a high concentration of ribosomes in there. Huh, I didn't even have to say anything. Were we in volleyball? You had to think about that. But if you need to watch, you're going to have time to do that. It's just, we'll wrap things up here shortly. All right. So does everyone have this content that wants it?
Yep.
Okay, so we're looking at 3 o'clock. Let's do this one, that one, and then we'll stop after that one. Then, yeah, maybe you can keep going, Mr. Okay, we can go ahead and do that if you would.
Yep.
And you're going to be a YouTube star.
Oh, awesome.
Okay, so if you just want to set it on here or clip that on your person, this on yourself or on your person. Okay, just no gang signs.
Okay.
No, that's only one stipulation.
Alrighty, I can handle that. I gotta figure out which one does what.
So if there's an appropriate term for this, we call it flying blind. Okay, but Mr. Wagner has had enough biology that this is very basic and can probably think of better examples than myself to explain some of this content.
Yeah. So next we're going to talk about vacuoles. So what vacuoles are is basically the storage containers for cells. It stores a variety of materials and waste products. So let's think about fat cells. Fat cells would be a good example of something with a large vacuole. The reason they have such a large vacuole is so they can store fats and then be able to use that at a later date when you need that chemical energy from the food, basically. Also, waste products, yeah. So if, let me think of an example for waste products that a cell would create. Maybe water. You could, you know. I take that back. I can't think of anything off the top of my head. All right, next we'll move on to lysosomes. Lysosomes are the organelles inside of a cell that break down other worn out organelles. So something that a lysosome would break down, some of the parts would go into a vacuole for storage. So that would be an example of a waste product. So a structure that breaks down worn out organelles and food particles. It will also break down bacteria and viruses that may enter the cell. So that's after the viruses and bacteria get past all of your other immune defenses. Once inside the cell, the lysosomes will try to take care of that before the virus or bacteria actually kills the cell.
So that's like the cell's inner defense mechanism.
Yeah, it's the inner defense mechanism. So like your immune system is your outer defense mechanism, and inside the cell is the lysosome.
So your white blood cells don't do their job, or they do.
Right.
And those get inside the cells.
Yes, so if the white blood cells miss something, then lysosomes will take care of it, or will try to at least.
They can't take over more cells.
Right, exactly. Yep. They can fuse with vacuoles and break down waste stored inside. Okay, so I don't know, pretty simple. If there's large waste products sitting inside those vacuoles and the cell doesn't quite know what to do with them, they'll send in lysosomes and break them down into further pieces until something can transport them out the cell and into the bloodstream. Okay, cell structure. Centrioles. So centrioles are similar to the cytoskeleton. These structures will function in cell division. Oh man, I gotta remember what these are. Oh, okay, fantastic. Found near the nucleus. Yeah. That doesn't help me a whole lot because there's centrioles and then centromeres. I forget this. We'll circle back when Mr. McKinney's back. Oh, cell structures, the mitochondria. I know this one pretty well.
Powerhouse of the cell.
It is the powerhouse of the cell. Thank you. It really is. Am I in your way? Just let me know. It will take substances within the cell and produce energy. So that's simple enough.
Is the mitochondria the ones to produce like the most waste in the cell?
You know, it might be. So what it does is you breathe in O2, right? And you need the O2 and the H2O, which is water, so oxygen and water. Your mitochondria will take in both of those and, is it sugars? It might be sugars. It'll take in all that and it'll break stuff down. So it'll create CO2, I think. Yeah, CO2 and other waste products that will go to vacuoles. So yes. In other words, yes, I had to walk myself through that one. Set of folded membranes inside to increase the surface area for breaking bonds. So like I was saying, needs the O2 and the H2O in order for the full, what do you want to call that? The full cycle to complete. And what you'll be left with is something called ATP. This might be.
Oh, we know a lot about
You know what ATP is?
Mr. Williamson.
That's right.
Preaches on ATP and track.
Okay. Well, I'm glad you know about it.
Don't even worry about it.
Yeah.
Like, that will forever be set in my mind.
Perfect. That makes it easy for me. So
Got 2 minutes to fill up your ATP.
inside the mitochondria, you will have ADP, and it'll convert into ATP. So it'll add another, adrenotriphosphate, so it'll add another phosphate. Energy collected from breaking bonds is stored within the structure, so meaning ATP.
And then that goes to the cells.
Yes, that goes to the cells. So basically what ATP does, I might be getting off subject, but I'm not sure. What ATP does is then, so you have ATP, and it's at a higher energy state, you can call it, than ADP. So it goes to whatever needs it in the cell, and that will take the ATP, break the bond on the ATP, and which will cause the ATP to go down to ADP, and that energy released from that bond breaking will go into another process within the cell. And that's kind of how your cells run. All right, we will go to the next one, chloroplasts. Yeah, absolutely.
And then, Mr. Bacon, when you're done.
Mr. Bacon.
It's all right. I understood.
I was just talking to Mrs. Preheim about Mr. Deitch's student teacher last year, which was who? Mr. Bigeman. So I had that name in my mind.
That's all right. That's all right.
And Mr. Wagner, when you're done,
Yep.
just go ahead and hit the red button for stop.
Okay.
Just close the laptops and or lock it, whichever,
Yep.
and I will upload that video upon my return.
Sounds good. All right. See you next week.
Yep.
It's not.
Okay, good.
And how far it is that you want to go, I would say mitochondria, chloroplast. Okay, I think that would be a good stop right there.
Right here?
Cilia and flagella.
All righty. Perfect. So we'll finish this up. So it looks like stop right now or finish this one?
Finish that.
All right, perfect. Okay, good. So like I was saying with mitochondria, chloroplasts are the same, well, not the same, but they have the same idea as mitochondria in animal cells, but they're in plant cells. So chloroplasts what do plants do? How do they make their food? Photosynthesis. So all the photosynthesis is done inside the chloroplast of a plant cell. So structures are used to capture solar energy. Conversion to chemical energy takes place because of photosynthesis, like we just said. And then thylakoids is where the process takes place. So, oh man, how in depth do I want to do? So, That's a good point. It does. Thylakoid. Yeah. So what it does is you have the overall structure, the overall chloroplast, and then inside the chloroplast are the thylakoids. So they're, where's a marker? Here's a marker. Okay, so chloroplast, I don't have a lot of room, so sorry. And then inside are these structure like this. It kind of looks like this, and there's multiple of them. And then the light comes in and hits the thylakoids, which, well, it's been a while since I've had that class, but which basically cause the same sort of mechanism instead of using O2, they use CO2 and release O2. So they take a carbon from the air and add it to probably a chain, if I'm remembering right. And then they're able to use that for later. So
with plants, they're
yeah,
like their cells directly take in sunlight, unlike our cells.
correct.
They take it from-
Yeah, so think of it as the exact opposite. So you know how mitochondria take in food and water and O2 and create energy, right? Well, chloroplasts take in light and CO2 and water and create food. So that's how they make plant food, which can be stored for a later date for themselves. Kind of like
Chloroplasts and mitochondria, are the same exact thing, but they do kind of the same thing.
they do the same purpose, but
They take in different things.
how they do it, how they take in different things, and their byproducts are different. So that'd be the main thing. All right, cell structure of the cell wall. This is an easy one. This is only in plants again, or not only in plants. It's in plants and bacteria and viruses because, plants, I don't know why it is in plants, but it probably keeps the rigidity of a plant so it can stand straight up in the air without relying on other mechanisms to do so. So that cell wall, it's also for protection. So instead of just a plasma membrane there's a hard, rigid structure that they can use to prevent infections and stuff like that. Associated with plants, thick, rigid mesh of fibers, and provides a rigid amount of support to stand upright. Okay, so just what I said. All right, we'll finish up with this, cilia and flagella. Some cells may have these projections outside the plasma membrane. This provides movement for the cell. So, marker. I'm going to take this. Okay, you have a cell, and then, okay, I'm going to do that a little differently. Yeah, you're probably right. It's a great idea if I know how to do it. Hey, look at that. All right, so we have a cell that looks like this, and it'll have a little tail. That should be the flagella. I can't remember. It's cilia and flagella. It's been 3 years since I took a bio class, so I'm really working on it. I didn't get a refresher today. So this is a flagella, and then with another cell, you'll have kind of hairs coming out like this, and that would be the cilia. Thank you. Yeah, it's kind of similar to that. And they kind of... So these are two cells is what they're supposed to be.
Anybody ever find that back on my
A cell.
He didn't do it for that. He had his mic off.
Are you kidding?
Oh, the sus?
No.
Yeah, that's what you're talking about? Oh.
No, like 3 days ago. he said something really. I don't even.
Preston, you can say. He said Steven.
Oh, he said screaming, and then John started laughing, and then he said it like 8 more times when he was screaming, and then he smacked that book, and every smack there was dust
Said the word.
flying off of it.
That
and then
happens.
we all started laughing.
Yes. Semen will use flagella. Semen will use flagella.
His exact word was, now let's take a look at the bovine semen industry.
I think that's it.
Standing